Hello everyone. In this video I am going to discuss about histological features of epithelial dysplasia. Okay, as we all know, epithelial dysplasia is a histological form of leukoplakia, and leukoplakia is a pre-malignant lesion or condition where we will see certain changes in the histology of a cell or epithelium. Now let's discuss about these changes, what we can see in the epithelial dysplasia. Okay, so in epithelial dysplasia you have two types of changes. They are architectural changes, cytological changes. First let's see about the what are the what are the layers of an epithelial membrane or the layers of a mucous membrane. So the normal layers of the epithelium there are four types. The first one is called as stratum basale. The next one is called as stratum spinosum. The next one is called as stratum granulosum. Topmost layer is called as stratum corneum. These are the four normal histological structures that are seen in the epithelium or oral mucosa. Now, first let's see about the architectural changes. The first architectural changes where you will see is irregular epithelial stratification. So, normally here you will have two layers of stratum basale, 2 to 4 layers of stratum basale, 2 to 3 layers of spinosum, 2 to 3 layers of granulosum, and top layer of corneum. Whereas here what happens in irregular epithelial stratification, you will not see these 4 layers one by one another. For example, stratum basale can be seen somewhere in the stratum granulosum can be seen somewhere in the spinosum can be seen somewhere near the corneum. So there will not be 4 distinguished structures like basale, spinosum, granulosum and corneum in the epithelial dysplasia. That is your first one. Next, second one is loss of polarity of basal cells. What do you mean by polarity of basal cells? Usually here in the stratum basale, this is your base membrane and this is the basal cell. So at any time this basal cell should be oriented perpendicular to the stratum basale, or the cells of the stratum basale should be placed always perpendicular to the base membrane. This is called as polarity. What happens in the dysplasia, there will be formation of the cancer cells due to which this cell will no longer be present perpendicular to the base membrane and the cell will be seen somewhere floating outside the base membrane. This is called as loss of polarity. So in the loss of polarity, the cell will be seen somewhere like this or somewhere like this. It means that it keeps detaching from the basement membrane. That is called as loss of polarity. Next, the third one is basal cell hyperplasia. What do you mean by basal cell hyperplasia? Usually what I have told, the stratum basale will be 2 to 4 layer thickness in the normal oral mucosa, whereas in basal cell hyperplasia there will be more than 2 to 4 layers of cells, might be 4 to 8 layers of cells. And why this happens? Because here the tumor cells or the carcinomatous cells, they keep proliferating. So because they keep proliferating and most of the proliferating cells in the mucous membrane is present in the stratum basale, that's why this layer keeps on increasing, and that's why the first major change we will see here is basal cell hyperplasia. Next one is drop shape retipex. Normally the retipex are present like this uniformly. They are attached to the underlying connective tissue. What happens in the drop shaped retipex? So these are the normal cells, right? These are the normal basal cells. What happens here? These basal cells, they keep proliferating. The basal cells, they keep proliferating and they keep trying to invade into the underlying connective tissue like this. Okay, so they keep trying invading into the underlying tissue by proliferating downwards. That's why you will form drop shaped retipex. Okay, next one. What is the next point here? Increase the number of mitotic figures. So normally in the stratum basale only you will see mitotic figures. That too 2 to 4 mitotic figures are common. Why? Because in the stratum basale you have cells that keeps on proliferating to form new cells. That is your normal structure. But here in the dysplasia you will see increased number of mitotic figures. Like mitotic figures will be seen in all stages of the cells. You will see mitotic figures increased number in stratum basale. The reason is same. Here the cells are proliferating repeatedly, due to which there will be new formation of cells, which indicates that there are formation of mitotic figures. Next, sixth one is abnormal superficial mitosis. As I have already told you, mitotic figures are seen only in the stratum basale, that is basal and parabasal structures only you will see mitotic figures. But in the dysplasia, these mitotic figures can also be seen in the superficial cells like granulosum or corneum. Why? Because already we are discussing these are the dysplastic features, means here carcinoma cells are keep on proliferating, so they will not see from which layer they are proliferating. Slowly these cells, the stratum basale proliferating cancerous cells, they will slowly separate from the basal membrane, they will go into the granulosum, they will proliferate, or they will grow into corneum and they will proliferate. Like that there will be proliferating mitotic cells in the superficial layers also. Next one, premature keratinization in the single cell. Usually keratinization occurs in the corneum. Okay, usually keratinization occurs in the corneum. That's why keratinization in the corneum is a normal structure. But premature keratinization means the keratinization will be seen in the spinosal cells. There will be formation of reddish keratinization surrounding the cell. This is called as premature keratinization. Next, keratin pearls within the retiridges. What are these keratin pearls? Usually keratin pearls are seen, usually keratin is seen in the corneum layer. Here only you will see keratin, parakeratin or orthokeratin. But what happens here, within the retiridges you will see keratin formation. What do you mean by this? Keratin formation is the structure of the, actually it is the function of the squamous cell to produce keratin, and this will be usually present in the corneum. But here a carcinomatous cell, it will also loses its function and it will produce excess keratin, which leads to the formation of keratin in the retiridges only. Okay, these are the architectural changes. Next one is cytological changes. What are the cytological changes? Means here the changes are present in the individual cell. Abnormal variation in nuclear size. Usually the nuclear size is 1:4 ratio. But here in one cell there will be 1:1 change in the nucleus. Here there will be bigger nucleus, here there will be more bigger nucleus, here there will be smaller nucleus, here there will be other bigger nucleus. So you can see several types of nuclear sizes in one cell, in one layer of cell, that is called as abnormal variation. Next one is abnormal variation in shape. Usually the nucleus is round in shape, but in some places you can see kidney shape, some cases you will see star shape, some cases you will see big round shape. So you will see several types of nuclear shapes. Third one is abnormal variation in cell size. Usually the basal cells all are same size cells, whereas what happens here, you will see one small cell, you will see one large cell, you will see one abnormal cell. So there will be different types of sizes of cells. All are these are due to the carcinomatous changes in the cell. Next one is increased nuclear cytoplasmic ratio. So normal nuclear cytoplasmic ratio is 1:4. Means if the cell is 4 times, the nucleus is 1 time. Whereas here increased nuclear cytoplasmic ratio means the nuclear size keeps on increasing and it will changes to 1:1 in dysplasia. Okay. Next one is increased nuclear size. As we have already discussed that this nuclear size will be increased. Next one is atypical mitotic figures. As I have already told you, atypical mitotic figures means mitosis will have certain stages. Atypical mitotic figures means here you will see different types of unidentifiable mitotic figures in several layers of the cell. Some may be star shaped, some may be in the chromatin structure like, some may be in the dividing form. So different types of mitotic figures are seen. The next one is increased number and size of nucleoli. So what is a nucleoli? Nucleoli is another small structure that is present in the nucleus, that is called as nucleoli. So what happens here? Nucleoli is the main part in protein formation. Okay, so a cell to divide, it requires more amount of protein. So the nucleoli along with the cell, the nucleoli will also divide and gives the cell the important requirements to grow. That's why increased nucleoli means it is rapidly dividing cell, which is again a character of dysplasia. So these are the histological features of epithelial dysplasia. Thank you.